Hello and welcome to Teaching Bio. Today we're going to be looking at aerobic respiration in under 15 minutes for A-level biology. Okay, so to start off with, here I've got a picture of the mitochondria and we'll go through and identify what each of the structures are. So firstly, within the mitochondria we have 70 S ribosomes. Now these ribosomes are important for protein synthesis, okay? And protein synthesis is very important in order to produce the necessary enzymes and other proteins and coenzymes and channel proteins and carriers that are needed to allow for the steps of respiration to take place. So we also have enzyme molecules that are bound, okay, to the inner membrane, and these will carry out the electron transport chain in order to allow for the production of ATP. We've got DNA, which contains the necessary information needed for the mitochondria to replicate, and also the DNA contains the necessary genetic sequence to allow for transcription and translation, therefore allowing for protein synthesis. Nextly, we have the inner membrane. We've got the intermembrane space and the outer membrane. So the outer membrane controls the sort of entry and exit of materials such as ions and other compounds and proteins in and out of the mitochondria. We've also got the intermembrane space, which is important to allow for the proton gradient to be maintained. Next, we have the cristae, which contain, again, the necessary proteins involved in the electron transport chain. The cristae have a large surface area, meaning that there is a faster rate of the electron transport chain, meaning that there's a faster rate of the production of ATP. Next, we have the matrix, which is sort of the fluid base space within the mitochondria, and this contains the necessary enzymes and other proteins involved in the link reaction, the Krebs cycle. It's important that we have many enzymes and proteins packed within the matrix to allow for a faster rate of ATP production. So it's important to understand and be able to identify firstly the features within the mitochondria and do this on a picture or an electron micrograph, and then to take it a step further, you could be asked in an exam to actually apply your knowledge and look at why each structure is important and how that helps with aerobic respiration and ultimately the synthesis of ATP. So we're going to take a look at the different steps in aerobic respiration, and firstly we're going to start off with glycolysis. The important thing with glycolysis is that glycolysis occurs in the cytoplasm, and they will love to ask this question in exams. So if we go through the different steps of glycolysis, glucose undergoes phosphorylation via two ATP molecules which are hydrolyzed into 2 ADP and pi using the enzyme ATP hydrolase, okay? So if we look at the picture at the bottom, we've got the glucose molecule there. 2 ATP are going to be hydrolyzed into 2 ADP and pi, which is going to release energy, and this is via ATP hydrolase. This will then produce some phosphorylated glucose here, which I'm just going to put down as P-glucose. The phosphorylated glucose is more reactive and has a lower activation energy due to the presence of phosphate. This then breaks down into 2 molecules of triose phosphate. So we've got 1 molecule of the 6-carbon glucose and 2 molecules of the 3-carbon triose phosphate, okay, which makes sense if you think about maths. 6 times 1 is 6, 2 times 3 is 6. Now, the 2 molecules of triose phosphate are each oxidized to form 2 molecules of pyruvate via the transfer of electrons from the triose phosphate to NAD, which will then form NADH, okay? So an important thing to remember with all the steps here is oil rig, which you may or may not have heard of before. But oil rig is basically oxidation is loss of electrons and reduction is gain of electrons. So as we are forming reduced NAD, reduction has occurred in that the NAD has gained the electrons from the triose phosphate. In this step, there is also the combining of 4 ADP and pi molecules to form 4 ATP for a condensation reaction by substrate level phosphorylation. Therefore, overall, we have a net gain of 2 ATP molecules because we put in 2, we get out 4, meaning that there's a net gain overall of 2 ATP, and it's important that you can recall that. Okay, on to the next step. So we're left with 2 molecules of pyruvate because pyruvate is a 3-carbon molecule. So remember, 2 times 3 is 6, glucose is 6 carbons. So the 2 molecules of pyruvate will then enter the mitochondrial matrix by active transport, and that will need a bit of ATP. Then the link reaction occurs, and this reaction is going to happen twice. Okay, we've got 2 lots of this reaction happening. So each pyruvate molecule is firstly oxidized, further reducing NAD to NADH. Remember, oxidation is loss of electron, reduction is gain. So in this way, the pyruvate is being oxidized because it's losing its electrons. So the electrons are being lost from the pyruvate, and the electrons are being gained here instead. Okay, so we've got loss of electrons and gain of electrons. The same molecule will then undergo decarboxylation, which, as the name suggests, is the removal of carbon, and that will then form acetate, which is 2 carbons and also carbon dioxide. So we've got 2 carbons here, 1 carbon there, 3 carbons in total, but we started off with 3 carbons. Then acetate is going to combine with coenzyme A to form acetyl coenzyme A, which is then going to be used in the Krebs cycle. Now this happens per 1 molecule of pyruvate, therefore at the end we are left with 2 lots of acetyl CoA for the 2 pyruvates that we had initially, and we give off 2 lots of carbon dioxide, and we have 2 lots of NADH being formed from this step here. So let's look at combining the numbers and what we had, what did we form, and what we are left with now. So for glycolysis, 2 NADH, 2 pyruvate, 2 ATP. The link reaction, 2 further NADH, 2 carbon dioxides, and 2 acetyl coenzyme A's. Therefore, in total, we are left with 4 NADHs, 2 acetyl CoAs, 2 carbon dioxides, and we've produced 2 ATP. So it's important to remember that the link reaction does not actually produce any ATP. Rather, the glycolysis reaction produces ATP via the net gain that we talked about before. So then we move on to the Krebs cycle, and this cycle happens twice because this cycle occurs for every 1 molecule of acetyl-CoA, and as we talked about, we've actually produced 2 molecules of acetyl-CoA. So everything in this cycle will happen twice per 2 molecules of acetyl-CoA per 1 molecule of glucose. So once 1 glucose molecule comes along, it's going to form the 2 acetyl-CoA because of the 2 pyruvate being formed, and this cycle will happen twice, meaning we're going to get 2 lots of every product that we form here. Okay? So if we take a look at the individual steps, you do not have to recall the names of the compounds here. However, it might be a good idea to learn the names because it makes it a bit easier, and you can always use these in your essays if you're doing AQA A-level biology and get some further marks. So if we look at stage 1 of the Krebs cycle, firstly the 4-carbon compound oxaloacetate undergoes carboxylation to form the 6-carbon compound citrate via the 2 carbon atoms from acetyl-CoA. So acetyl-CoA, again, 2 carbons, comes along with +2, 4 + 2, we get 6. This then actually produces coenzyme A, which is left out, and this can actually go back and then be recycled back into that link reaction to form even more acetyl-CoA. Stage 2. So we're on 6-carbon compound citrate. This then undergoes oxidation by reducing NAD to form 1 molecule of NADH. Okay, so the NAD is going to gain the electrons, and the 6-carbon compound citrate is going to lose its electrons. Citrate then undergoes decarboxylation to form 1 molecule of carbon dioxide and the 5-carbon compound alpha-ketoglutarate. Then alpha-ketoglutarate will undergo even further oxidation, reducing more NAD to form another molecule of NADH. And in this process, 1 molecule of ADP and pi are condensed to form 1 molecule of ATP via substrate level phosphorylation, okay, which is occurring there. So it's important to know that ATP formation in the Krebs cycle will occur between the transfer of the 5-carbon compound to the 4-carbon compound. Alpha-ketoglutarate then goes further decarboxylation, removing 1 molecule of carbon dioxide, and then forming the 4-carbon compound succinate. So we've got another molecule of carbon dioxide being formed. Stage 4, we go from succinate to fumarate. Again, further oxidation, and this time FAD is actually reduced to form FADH2. and then succinate is converted into 4-carbon compound fumarate via the oxidation, and this obviously requires the use of enzymes, as all the steps do. Then fumarate undergoes structural changes to form the 4-carbon compound malate. Again, you don't know these names, but it might help you recall them. Malate then undergoes oxidation to reduce NAD to produce 1 mole NADH, and then generate again from the start the 4-carbon compound oxaloacetate. So it's the Krebs cycle, so we've repeated the cycle, and then that will happen one more time. So if we total up what we produce from the Krebs cycle, once it's spun around twice per glucose molecule, we've got 2 ATP, 4 carbon dioxide, 6 NADH, 2 FADH, and we've also got these products left over from before. So overall, we have 4 ATP, 6 CO2, 10 NADH, and 2 FADH. And we need to produce the remainder of the ATP by oxidative phosphorylation, okay? So this ATP was produced by substrate level phosphorylation, and this ATP was produced by oxidative phosphorylation. It's important that you can remember the distinction. How is the remainder of the ATP produced? Well, it's produced via the electron transport chain, which is this beautiful process outlined in this picture here. So we'll go through the different steps. So firstly, if you recall, we produce NADH and FADH2. These are actually known as reduced coenzymes. So what happens this time is the reduced coenzymes are actually oxidized. Okay, so they are full of the electrons, and this time they are going to actually lose their electrons, and they pass them on to electron carriers. The electron carriers accept the electrons, and they basically pass the electrons on in a series from one electron carrier to another, like so. So they pass it from one to another, therefore there is this sort of continuous fluctuation of reduction and oxidation reactions down the length of the electron transport chain. So the moment it gains the electrons, it will obviously become reduced, and then the moment it loses the electrons, the same compound will be oxidized, and then the process will repeat as we go along. As the electrons are transferred from one electron carrier to another, there is the gradual loss of energy. So again, if we look back at this picture here, this is what I'm talking about, electron flow. They move on to these mobile electron carriers, and then energy is lost from here. So what's the importance of that energy? Well, that energy is actually used to pump protons, so H+ ions, out of the matrix into the cytoplasm to increase the concentration outside. Okay, so the proton pump, if I go back again, the proton pump is here. So all the protons here need to be pumped out there. And because they're going to be moving against a gradient, they need some kind of energy, and that energy is supplied by the gradual loss of energy via the electron transport chain. So they're pumped out to increase the concentration outside, and therefore they can diffuse back in, because we have a high concentration outside and a lower concentration inside. So once we've raised the concentration outside via the pumping, these protons will then pass back down, okay, through ATP synthase, and they'll bind to the allosteric site of ATP synthase to alter its structure and therefore yield the production of ATP. Okay, so this picture here sort of nicely summarizes where the different types of phosphorylations occur. Citric acid cycles within the Krebs cycle, and remember that glycolysis occurs in the cytoplasm. Finally, how is water produced? Well, at the end of the electron transport chain, the electrons are passed onto an oxygen molecule. Oxygen acts as the final or terminal acceptor of electrons. It's very important that you remember this statement. So electrons combine with the oxygen, and then there's 2 free hydrogen ions because of the massive pool of protons we've got, and that then forms water. And here's the entire process summarized, and there's sort of ATP synthase, there's the proton moving down. So, just to finish off, we're just going to talk about respiration of lipids. Lipids are hydrolyzed into fatty acids and glycerol by lipase. The glycerol can then be phosphorylated and then go on to form triose phosphate, so it can re-enter the cycle. Then that can go on to form acetyl-CoA, as we talked about before. Fatty acids can directly be broken down to 2 carbon fragments, and that can be put into acetyl-CoA for the Krebs cycle. And then finally, respiration of proteins. Proteins are hydrolyzed into the amino acids via sort of peptides. We'll remove the amine group by this process called deamination. That then just leaves the carbon chain, and then that carbon chain can enter the Krebs cycle and so on forth. Okay, so pause the video and have a go at this question, and the answer will follow. So here's the answer. This is a very common question. It's the fact that glycolysis occurs in the cytoplasm. That's the key statement here. Okay, so pause the video and have a go at this question. Okay, and here